And first, I'd like to thank the HFSP for funding this and allowing this wonderful cooperation. You see, it's a collaboration between Walter Federle, whom you see here on the left. That's us having breakfast a few days ago. Fumitoshi Kaneko from Fukuoka, and myself, and I'm from Mainz in Germany. Yeah, hydrocarbons are basically a multifunctional suit. So they form a waxy layer on the body surface of all insects. And when you think about you put on sunscreen, when you put on sunscreen on your skin, this layer of sunscreen will be still several times thicker than the cuticular hydrocarbons, so it's extremely thin, as we'll see in a moment. What is funny about them is that the composition is incredibly really diverse, and it's highly species-specific. So it's diverse within the same insect, but it's also extremely diverse across insects. What you see here on the right is monograms of two hydrocarbon profiles. The black one is the red garden ant, Myrmica rubra, and the red one is Myrmica ruginotus. It's its sister species, so they couldn't be much more closely related. And you see they are quite different. There are some substances in common, but most are actually different. Myrmica rubra is mainly characterized by analkanes, monomethyl, dimethyl, trimethyl alkanes. So you see hydrocarbons are actually quite simple molecules. They're just chains of carbon with bonded to hydrogen, and sometimes they have these methyl branches. And in Myrmica ruginotus, they're a bit different. They also have these branched hydrocarbons, but they also have alkenes and alkadienes, so it's a carbon chains with one or two double bonds. The methyl groups and the double bonds can be at different positions, and also the chain length varies between 20 and 40. So altogether, you can have 50 to more than 100 compounds on a single insect. To give you two more examples, this is a desert locust, which is one of our model systems. And what you see here on the x-axis is the length of the molecule as the number of carbon atoms. And you see that most of that is actually the red. It's N-alkanes, so unbranched alkanes. And there's also some branched alkanes with one or two methyl groups. Another model system of ours is the American cockroach, where more than 70% is just a single compound. It's this alkadiene. And then there's a methyl branched alkane and then an analkane. I just wanted to introduce how incredibly diverse that is. And sometimes, so you can use this cuticular hydrocarbon profile actually as a fingerprint. You can use it to disentangle sister species, which are just so genetically different, but chemically it's very easy to tell them apart. So that makes them very, very, very convenient. What I mentioned is they are a multifunctional suit. So they have a lot of different functions, and that makes them so exciting. So, for example, as you see here, they serve as chemical communication signal. They carry information. When two ants, for example, meet, they touch each other with the antenna, then they smell each other's CHC profile. CHC is cuticular hydrocarbon. And then they know, okay, the other one is male or female, or queen or worker or larva, an inside worker, outside worker. She's healthy or she's not healthy. She's from a different species, my species. She's from my nest or a different nest. And all that information is encoded in the CHC profile. So that's CHCs are pretty much the most important communication cue. They are also important for sexual selection in many different insects, in beetles, flies, whatever you want. And they are also important for waterproofing. So this thin film of CHCs keeps the insect from drying out, which is especially important while outside when it gets hot and dry. And we'll see that in a minute. What is much less studied is they are important for foot adhesion. So CHCs are sometimes secreted by the tarsal pad on the feet, and they help bridge gaps between the surface and the foot, and this enhances foot adhesion. So when the insect wants to climb up the stalk of a plant, they often need CHCs in order to be able to do that. They also serve as lubrication to lubricate joints when the insect moves, and they can also self-heal. So when you scratch the CHC layer, it closes by itself without any enzymatic help. The question is, how are these functions determined, and might understanding what the CHC layer needs to do help us understand how this diversity involved, and actually how can such a layer fulfill so many functions at the same time? And we figured that the physical properties may be key to that. So, for example, when you look, what do you need? for chemical communication here. You need signals that have a low viscosity, that are ideally even volatile, so that they are readily accessible to the other individual. For waterproofing, you actually have quite the opposite. You want substances with quite a high viscosity, or even solid phases that prevent water from diffusing out of the insect body. For food adhesion, you need somewhat of an intermediate viscosity, so it shouldn't be too low, it shouldn't be too high, to bridge this gap between surface and food. The surface tension of the CHCs is important. For lubrication, what counts there is the thickness of the CHC layer, the viscosity and its phase behavior. And for self-healing, also fluidity is very important. So the research questions of our HFSP projects are, first, okay, what is actually the structure, the physical structure of the CHC layer, and what are its properties in terms of phase behavior, viscosity, and spatial heterogeneity? Then we wanted to see how can it fulfill so many functions. First, how do these physical properties actually affect the performance in the different functions? And then how does temperature affect all these physical properties? I mean, most physical traits are affected at some way in temperature. So do insects adjust, take account of that? Do they modify their CHCs to adjust when the temperature changes? And then how does the CHC composition, which is species specific, affect these physical properties? And by understanding that, can we then explain maybe why such a CHC diversity evolved? Yeah, so first, let's look at the phase behavior of pure cuticular hydrocarbons. So if you have, for example, pure analkanes, they aggregate very tightly. So they have the whole chain can aggregate, pack very tightly. They form crystals. When you look at the CHCs between 20 and 40 carbon atoms, which is in the length that what insects have, due to van der Waals bonds, they crystallize and they are solid at room temperature. When you introduce methyl branches, you disrupt this aggregation pattern. So they can't aggregate as tightly, so at room temperature they are solid or highly viscous liquids. When you have more methyl branches, you increasingly have less viscosity, so the melting point goes down and they tend to be liquid at room temperature. And the same is true for alkenes or alkadienes. So from left to right, we have a decrease in viscosity and also in melting point, and then also in waterproofing ability. So alkanes are fantastic for waterproofing, alkenes are not. What changes with temperature when we now look at real CHC mixtures? And we did that actually together with Svenja Morsbar from the Max Planck Institute for Polymer Research. We extracted hydrocarbons from different ant species and performed differential scanning calorimetry. And here you see extracts of two different ant species. And what you see here is melting curves, so that the peaks basically show dissipated heat when you increase the temperature. So at each peak you see that something melted. And now when you look at that, you see that CHC layers start melting at minus 60 degrees already, so really low. And they continue melting until plus 40 degrees. So like here, plus 40, in the other species, everything is melted as about 25 degrees. So in all that temperature range, you have a constantly changing mix of solid and liquid phases. So it's a very complex phase behavior. And by the way, this was not only measured in extracts, but Fumitoshi could also confirm that by measurements in situ on insect wings of cockroaches. Now when we focus on the viscosity of the liquid phase, let's only look at the liquid phase. Sorry, I forgot that. You actually can see solidification. What you see here is a droplet of cuticular hydrocarbons, and you see these tiny structures there, and they form only when you cool down the CHCs. So at warmer temperatures like 25 degrees, it's all liquid. When you cool it down, you see sort of these crinkles emerging, and that is an indication that they solidify. So they form a sort of gel when temperature goes down. Yeah, so now let's look at the liquid phase and measure the viscosity. And we did that using microrheology, which is a method. You have a droplet of a liquid and you insert tiny beads, and then you basically observe the Brownian motion of these beads, and this allows you to infer the viscosity. We did that together with Béranger Abou from the Université Paris-Cité and the CNRS. And we did that for hydrocarbons of a number of ant species. And you see that they are actually quite homogeneous. At first, we thought that at least. But the viscosity of most hydrocarbons is in the range of motor oil or olive oil. So it's much more viscous than hexane or water or milk, but it's less viscous than honey. What Selina, our PhD student, now discovered is that it's actually biphasic, which is quite cool. So you have, the liquid phase is not homogeneous. You rather have a mix of oil-like hydrocarbons, so with the viscosity of oil, but you have a much smaller peak with honey-like viscosity, so much more viscous. And this is separate, so it never really mixes, which is quite cool. And we think this is because structurally similar CHCs vary across samples. So this is now, as an example, I put the CHCs of the desert locust, but we found that in a range of other insects again. And you see here all the different hydrocarbons that we found in the desert locust. And we looked which of these molecules co-vary across different samples, across different extraction treatments or extraction techniques. And what you see is we found strong covariation among CHCs that are structurally or physically, probably then also physically similar. For example, we have covariation in these longer analkanes, we have covariation in the shorter analkanes, and interestingly, we have this cluster with nearly all of the internally branched in the monomethyl and the dimethyl alkanes. So these are the ones that have a methyl group, either two methyl groups or one methyl group, which is sort of in the interior on the molecule and not towards the end of the molecule. And it's quite likely, or at least possible, that these co-varying substances form clusters of a certain viscosity, and then there's others that also co-vary and sort of aggregate in the liquid phase to form clusters of higher or lower viscosity. Yep. So is there a spatial structure? And to elucidate that, Fumitoshi did some measurements with optical photothermal infrared spectroscopy. It's called MIRAGE. And that's a technique where you have a tunable pulsed mid-infrared laser that heats up, basically heats up the object at a certain frequency, and that induces photothermal expansion. And this you can measure then with an optical laser. And this enables us to determine the thickness of the CHC layer with a spatial resolution based on the signal from CH2, which is the bonds that you get in CHCs, and the NH, which is the signal that you get from the underlying chitin. And so what you see here to the upper left is the signal from the cuticular hydrocarbons from the CH2, and upper right is from the chitin, which is the NH bonds. And when you combine them, you see the lower picture, which and you see the resolution. So this quadrat is 50 micrometers long, and you see that there's very strong cell-shaped structures with higher CHC thickness. So the layer actually has a spatial structure, and we confirmed that with microscopic, like normal through light microscopic images, that this is really along the cell borders. The CHC layer is much thicker. And Fumitoshi did now some measurements of X-ray scattering, and this allowed us, again, we looked at intensity changes of the NH bonds and the CH2 bonds, and that also allowed us to infer that the CHC thickness varies between 10 nanometers, which are actually only a few molecules thick, and 500 nanometers, and this is quite immense. That's a factor, so the thickness varies by a factor of 50. I think that's quite impressive. And this is a potential scenario where it's possible that we have sort of these mountains of CHCs connected with much lower layers, valleys of CHCs. Yeah, what happens now in different temperatures? So when it's warm, this is a problem for the insect, because as we've seen, CHCs, like anything, melts or becomes less viscous when temperature increases. So, which also means that water diffusion increases because the layer becomes more permeable. At the same time, we have a higher water vapor pressure. So, I mean, water evaporates more quickly when it's warm, nothing new. But so we have two reasons that both cause that the drought stress for the insect is much higher. So even when it's not exceedingly warm, but just like, say, 29 degrees or so, this actually is much more drought stress than if it's 20 degrees for exactly these two reasons. And so the insects need to counteract that, and they do that by adjusting their hydrocarbons to make it more solid. And this is what we tested out, and we have confirmed that for quite a number of insect species already. These are our two friends from the beginning, Myrmica rubra and Myrmica ruginodes, and we kept them at 3 different temperatures, 12 degrees, 20 degrees, and 28 degrees, each time for 3 weeks. And we see that when it's warm, you see a very strong increase in the relative abundance of these analkanes. Do you remember analkanes are rather solid? So these increase when it's warm. At the same time, other substances which are sort of disruptive decrease. So when it's warm, you have much less dimethyl alkanes in the one species, and you have less of the alkadienes in the other species. That's actually not just a matter of mathematics, because there's a big chunk of intermediate hydrocarbons that can be up to 30 to 50% of the CHC profile, and they didn't change at all throughout the different temperature treatments. So we see that the insects really adjust to the current temperature, and that allows us to test temperature-dependent functions. So we can now test, okay, we keep the insect at 28 degrees, and then measure functional performance at 28 degrees, and then measure functional performance at a mismatched temperature when it's too cool or too warm, and what happens then. Yeah, and for example, we measured water loss. This is what Monami, the PhD student of Walter, did. And she measured water loss in the cockroach. And at 15 degrees, nothing happened. I mean, they lost a bit water, but not too much. But there we couldn't find any effect of acclimation. Both acclimation treatments yielded the same water loss. However, when it's warm, you see a strong increase. So the cool acclimated ones lost a lot of water, whereas the warm acclimated ones did a lot better. They lost a lot less water. Okay, how about friction? I mentioned that friction and lubrication is not really well studied yet. So what we did here is we measured shear stress. That was again Monami's work, and we looked at the CHCs. And we looked at when you have CHCs, so when you have a normal native cockroach wing, the shear stress of two insect body parts rubbing against each other is quite low. When you remove the CHCs, it's much higher. We could also confirm that with a completely different measure using the cantilever of atomic force microscopy of AFM. And we did that with a 2-spotted cricket. And there we also found quite a shear signal. And interestingly, this differed between male and female crickets. And now if you look at the chemical composition, the male crickets have more unsaturated compounds. So they have more alkenes, and this leads to a lower viscosity of the CHC profile. And we think this might actually be a reason why the friction is lower in the male crickets. We also have some other individuals, some other species now. So tentatively, we think that the friction actually might be determined by CHC composition. Yeah, but friction is not only bad. So it's not only about lubricating and getting rid of friction. You also need that in order to adhere to a surface for food adhesion. We did that some years ago with the stick insects, where we kept stick insects at 15 degrees and at 27 degrees. And you see at very smooth temperatures, the contact area of the foot and the surface is high enough, so you get quite a good adhesion, which you see here on the left. When it's micro rough, so when you have tiny bumps, the contact area is much lower, and there the hydrocarbons are good. And the cool acclimated ones, they have more liquid hydrocarbons, and they had a better friction and better foot adhesion, because these liquid hydrocarbons were secreted from the feet and went down to and could close the gap between the surface and the foot to adhere the area. The warm acclimated ones, they had more solid CHCs and they did worse. But when you turn it around, when you now go to warm temperatures, you see the opposite. Again, adhesion to smooth surfaces is quite high, but now at micro rough surfaces, the warm acclimated do better, and this is probably because now the warm acclimated CHCs are liquid enough, whereas the cool acclimated ones are too liquid, so they cannot close these gaps. So we see here that acclimation actually benefits in both directions. So cool acclimated insects benefit at cool temperatures, warm acclimated ones at warm temperatures. So to sum up, we've seen that cuticular hydrocarbons are really cool, a really cool trait in insects because they fulfill so many functions. They are very complex within an individual and between species, and we always have a mix of CHCs that melt or solidify at different temperatures, and that leads to really complex phase behavior. We have solid phases, we have a high viscosity and a low viscosity liquid, and we also found that this is probably because different CHCs cluster and form distinct domains according to their physical properties, and also form a spatial heterogeneous structure. And we think that this complex phase behavior allows that the insects can optimize different functions at the same time. We also saw that phase behavior continuously changes with temperature, so insects have to adjust to current conditions, and the acclimatory changes benefit the temperature functions like water loss and food adhesion. And yeah, with that, I'd like to thank my collaborators, the students on the project, funding agencies, and again, really the Human Frontier Science Program for enabling all that, and you all for your attention. Thank you.